Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Welcome to MIT Faculty Forum Online. I am Pam Belluck, an MIT Knight Science Fellow from the class of 2008, and I'm a New York Times health and science writer, and I will serve as your moderator. Today's broadcast is sponsored in part by the MIT Federal Credit Union, MIT Professional Education, and MIT Sloan Executive Education. As a reminder, we welcome your questions during this chat. Alumni joining us via Zoom can use the Q&A feature found on your toolbar, and we will get to as many questions as we can. We're very excited to hear today from Krystyn Van Vliet, a PhD from the class of 2002. Professor Van Vliet is also Associate Provost and Professor in both the Department of Materials Science and Engineering and Biological Engineering. She began her service as Associate Provost in the fall of 2017 and has been on faculty since 2004. As Associate Provost, her responsibilities include Institute space management, non-federal sponsors of MIT research and education, and technology transfer. In the lab, Professor Van Vliet's group studies material chemomechanics, material behavior at the interface of mechanics, chemistry, physics, and biology. A link to her full bio can be found in the chat window. Welcome, Professor Van Vliet. In the brief time that we have with you today, we are hoping to hear at least a little bit about your current research and your thoughts on this very unusual year for MIT students and MIT's research community. Thank you so much.
Thank you, Pam. I'm very happy to be with all of you today, and thank you all for making the time with all the things that you could be doing right now to tune in and chat with us. So I thought what I would share is a little bit about how did I end up at MIT in the first place. Some of you who are here today have been former students of mine. Some of you have never met me. So I thought I'd give you a little background and then talk about what I do at MIT really. Pam gave the brief bio, but the day-to-day operations in normal times and also in these pandemic times require a little bit more explanation, and certainly open to any questions on that. And then give a sense of what's next at MIT, particularly as I think about the research and innovation path for the scope of work I'll share with you today that I'm very excited about in our lab. Okay, so first, how did I end up at MIT in the first place? Well, this is a view of the country of the US. If you grew up on the East Coast and you never left, you think about this as pretty much the map of the country and the world. And so I'm guilty of being one of those people. So I was born in New Jersey in the Garden part of the Garden State. And I lived there until I went to college. And I worked in various capacities. I had many jobs down there, but one of them that I really enjoyed was working on a farm. It happened to be a Rutgers agricultural extension farm. So it was part research. I did not participate in the research. I was there solely to ride on awesome machines like this. This is a potato harvester. And I think I really thought of farmers as really creative engineers and problem solvers. They knew how to build this equipment, repair this equipment, think about the crop rotation, and I just was all in. I really enjoyed it, even though it was a lot of hard work. Then I actually went to college in Rhode Island at Brown University in Providence. And I was doing experiments like this, and that's where I really fell in love with research. I had great faculty advisors there. They were fantastic mentors. And they really instilled in me this love of mechanical behavior of materials that I didn't know even what that meant. You know, this wasn't a potato harvester. This was a different kind of machine. This is a photograph of a high school student who got to do this in high school, but I didn't have that kind of high school experience. So I learned in college, hands-on, all in, worked in that lab for all the years and all the summers that I could until I went to grad school. And it was just a tremendous experience. I can't say enough about how great it was to do research as an undergrad and really take responsibility for the success or failures of your own little research project. And then I went to graduate school at MIT and really enjoyed that experience, continued learning about mechanical behavior from a tremendous community at MIT in multiple departments. Really enjoyed that experience, but didn't think I necessarily wanted to become a professor. Graduated right after 9/11, which was its own crisis for the country and the world, and considered several different career options, but in the end was fortunate to have the opportunity to first postdoc at Boston Children's Hospital, where I learned a lot of new things, much more than I gave back to the very patient supervisors that I had there, and then came back to MIT on the faculty, as Pam mentioned, since 2004. And I have been in Cambridge ever since, with some minor caveats and twists as my research program has grown. But the favorite thing I have at MIT is my research group. I love working with the folks in the lab, whether they're undergrads doing their very first research project, as I got to do when I was in their position, to very experienced research scientists who've worked with me for over a decade, and all the graduate student learning in between. So that's what's really getting me out of bed in the morning to do all the things you need to do to run a successful research group and teach classes and raise money and contribute to the Institute community life. It's really having a great research group to think hard on identifying big problems and then also tackling them through collaborations. So one of the collaborations that's developed over the past over 10 years now for my group is a team that I also co-lead in Singapore. So I have a team in Boston, I have a team in Singapore. They're 12 to 13 hours time different, depending on the time of year. Right now, still 12 hours time different. So that takes a lot of communication, a lot of collaboration, a lot of understanding different cultures, and a tremendous opportunity for me. Singapore is a small island country. The first time I flew there as part of an MIT research project, I had no idea where it was on the world map, but it has become a second home and a tremendous part of the research experience as well as my family life. So with that brief introduction, I wanted to talk about what do I do at MIT really? And there's two main buckets of work that I do at MIT. The first, as Pam mentioned, is I serve an administrative role, and this is a service opportunity. It's a time where you take parts of your career that you have to put on pause. So for example, I don't actively teach in the classroom. I record classes for what will be an edX class next year, but I don't get to interact with students because my day is filled with a lot of activities that help the rest of MIT run well. But I have enjoyed it very much, and I certainly enjoy working with our provost, Marty Schmidt, and learning from him on that. So my role has two buckets of activity in that administrative space. One is the campus, and this includes all our MIT buildings that are being built right now, such as the College of Computing in Design, or MIT Nano that just opened a couple of Octobers ago, or the new space in Kendall Square that's opening up with lots of activity and new housing for the graduate students. But it also involves our off-campus research sites, the properties we lease around campus, renovating the beautiful old building where my lab is that is 1900 vintage but still does cutting-edge research. So there's a lot of details in thinking about the space, both strategic and tactical for the Institute. And then the second part that I work on is called the Office of Strategic Alliances and Technology Transfer. This is a new office at MIT, developed out of the ideas that our Vice President for Research, Maria Zuber, and our Provost, Marty Schmidt, had on changes that the Institute needed to make in order to address the changing funding landscape and operations of how we do research at MIT. So they got input from lots of people at MIT and outside of MIT, and driven by this desire to serve the faculty better as they work on those engagements with the outside world. Could be a big company, a small company, a philanthropic foundation, another country. This office was set up, and I help lead that with directors who run the day-to-day operations and have quite a bit of expertise in technology transfer, strategic transactions, so that's the lawyer part, and corporate relations. So that's a great team as well. And I think what I've learned from this Institute service, yes, it does take time management, it does take dedication, but you need to find it fun. It shouldn't be a chore, and it has been fun for me despite all the problems that need solving. now and in the past, because I have great teams that I work with, both on the campus space side and the OSAT side. So I'm happy to answer more questions about that. But I thought what I really do at MIT is I get to do research. And then because part of the deal is you're in an MIT community, you should also help that community operate. I'm willing, and I also enjoy doing the Institute service. So what is my research? It comes from really trying to understand more about the world around me. And those of us who grew up as materials engineers, as I did, you just see the world of materials. Why is it made out of this? How is it made out of this? Could it be made out of something different or made in a better way? So you start to become very observant of the materials around you. And I got intrigued by how does the mechanical behavior of these materials, like how stiff or strong or resistant to breaking they are, relate to the chemistry of those materials, maybe their composition, maybe the pH around them, maybe the temperature that's speeding up a chemical reaction. So that coupling between the chemistry and the mechanics inside materials and material interfaces, including biological cells, became very interesting to me because it sounded hard, it sounded complex, it sounded like something I knew very little about. And what I wanted to do to answer those questions was do not only experiments or only computational modeling and simulation, but really try and get those two approaches to talk to each other. My PhD experience at MIT included both experiments and computation, so I became very comfortable with the idea that I could design better experiments if I could model them first, and I could understand my experiments better if I could simulate the outcomes, right? So that back and forth has been very helpful to me in connecting concepts that might not be so obviously connected. So what are some of those concepts? One is there's coupling between chemistry and mechanics between cells and the other materials they interact with. It could be another cell, it could be a drug, it could be a material you've engineered to try and make the cells do what you want for a medical outcome. So these biological interfaces are complex, difficult to model, and really intriguing to study. So that's really the inspiration for our work. But we also study complex materials, often polymer nanocomposites or complex polymers that have biotech applications, but maybe may never interact with a cell. So for example, I had a student graduate, or actually is graduating later this week, whose thesis was on designing materials that could behave just like the brain in terms of their mechanical deformation, but never be used for cell culture. And then he used that same understanding to help design better helmets that would protect the brain when under high rates of injury, such as an impact with another person or another helmet or another wall. And then a third area are materials that have no biotech application, but they help us tease out this coupling between chemistry and mechanics in still very important and interesting systems. And those could be fuel cells or batteries, materials that are used in energy. And here the coupling is between the mechanics and a special kind of chemistry called electrochemistry. Cells undergo electrochemistry too. We just don't always use the same language as we talk across these different application spaces. But I enjoy that breadth, and I'm very fortunate to have a group that gets to work on that with me. So take a deeper dive here. What I'm showing you here is some snapshots from a movie where I had a very talented PhD student who was willing to help answer the question with me. When you have a single stem cell, and this is a cell that is from an adult from your bone marrow, you and I can donate these stem cells to other people. If I just watch that one stem cell divide, does it give rise to cells that are just like itself, or is there some variation as the progeny or as that family grows? And the reason why we were studying that so carefully is because stem cells have been considered for cell therapy for many, many years. And these particular cells from your bone marrow called mesenchymal stem cells, it's a bit of a misnomer. They're not all stem cells. They're not all working as hoped or intended as cell therapy. So we wanted to understand maybe part of the issue is because when the cells are forced to grow outside the body, they actually start to diversify and become heterogeneous. So there's one cell, now it's divided and become 2, then 4, then 8, and so forth. So you see this doubling over here. Oops, sorry. get my show back. Over to the right, what you see is another colony of cells that are growing. So you're watching a mesenchymal stromal cell colony form. These are human cells taken from human bone marrow. And you can see now this colorful diversity. So what this graduate student did was to work with a team of undergraduate researchers and figure out ways to identify the cells by generation. So what you see here is that the oldest cells are yellow, the youngest cells are purple, and so you see that the transition from warm colors to cold colors as that colony grew. But when you stare at this a little bit longer, and of course we stared at it for a very long time, you start to see some other differences in the colony, like those larger cells look a little larger to you, right, the yellow cells. So you can show these data this way. This is called a lineage tree, and you watch the first cell at the top was red, if you recall. Then it gave rise to two twins. Those two cells gave rise to two twins, and so forth. So you have this lineage tree of progeny that came from a single cell. And up to this point, it looks pretty textbook. Cells divide, they give rise to two more cells, and so forth. Now, the width of this line is how big those cells get in the process of dividing. And once you get past day 4, what the student observed here and in many other experiments is that you had some cells that never divided anymore. So they stopped dividing and they just got larger and larger and larger, and they were left behind in that renewal of the cell population. And we've gone on to study and understand what that might mean in terms of being able to use those cells effectively towards cell therapy in a variety of contexts. So this work is published. This glyph here just shows you that we observe that heterogeneity of color and generation and size of cells in many different experiments, and we can quantify that through automated image analysis. But they all came from the bone marrow, and they may have in other experiments come from the brain. So we see that in cells that come from the brain as well. And part of the challenge in getting that heterogeneity to go away is to recognize what's too simple about the culture conditions that we use in a lab or in a hospital compared to these native tissue environments, whether it's your bone marrow or your brain or another tissue and cell source or organ. And so we've learned about that by thinking about these organs, these interfaces for the cells as complex materials. So this has got us into now moving towards how do I think about cells in terms of using cells as medicine, not just something to study in the lab because the heterogeneity in the cell population is interesting, but how do I harness it, control it, understand it, so I can take cells that could be from the blood for a cancer treatment or could be from the bone marrow for a cartilage repair of your knee, and put them into a patient so that they're able to act as little drug factories and repair the patient for me. And these cells could come or go from brain tissue or from blood or from bone marrow. It depends on the disease or the indication that you're trying to address. And we've worked on various aspects of these because the challenges are common, but the approaches and the understanding we need to have about cells and materials is specific to the cell source and the target indication or disease. So one thing we've learned is if you think about trees in a forest, you look at a forest, you know it's all the same type of tree, but there's this variation among the trees, maybe due to the age, maybe due to the local water or nutrient conditions. And you can still recognize the forest, but you don't think much about the heterogeneity. Well, that's very, very true in cell therapy. There's variation from person to person in the type of cells you have available to give for a cell therapy treatment and the ability of your body to respond to a cell therapy treatment. So we've developed several different technologies in collaboration with many really dedicated teams over the past over 10 years now to deal with that. There's a few different approaches, and we summarize them like this. If you think about the fact that you're getting cells from a human, and I've shown you that if you take the cells of interest and you try and expand them out of the body under current state of the art, you're going to get some heterogeneity in that population. So you could put them into the human and they could be safe, but they may not be efficacious. They may not have the effect that you're hoping for. But what if you develop a rather brute force, even if it looks elegant, approach to expand the cells, accept that they're going to be heterogeneous in their function, but then provide means to sort them to get just the cells you want to put them back into that patient. So that's a sorting approach, and we've developed those. But an even better approach, because you can see the inherent waste in this process, you're only using a fraction of the cells that you spent time and energy and electricity expanding, is to figure out, well, what is the source of that heterogeneity? If I understand that well enough, can I engineer an environment that would allow the cells to expand while still being functionally all the same so that they can go back into the patient? And so that's an approach that we sometimes call mechanopriming of the cells. So I've developed those approaches in collaboration with others in Singapore as well as at MIT. And the heart of one of the approaches I'll show you comes out of the lab of Jay Han, a collaborator of mine for many years in electrical engineering and computer science, as well as biological engineering at MIT. And Jay loves to concentrate samples. So we started working together on concentrating and separating these mesenchymal stromal cells of various different sizes and stiffnesses and functions, so that we could get to an approach like this, where you put a mixture of cells. Some are engineered to have high therapeutic value, others not, but you can run them through this spiral microfluidic device, naturally separate them on the basis of their size. And we can talk about how and why that works, so that you could then take the cells that you want that will lead to the good therapeutic outcome, package those cells and provide them to the patient. So that's an example that we have in clinical trials right now in Singapore, and we have lots of other applications and variations on that approach. But the whole goal here is to think about, I want to provide the right medicine to the patient. We know how to do that with aspirin or certain cholesterol drugs, but we don't know yet how to do that well all the time for cell therapy. There's some very exciting approved cell therapies, but we want to get the medicine right for each patient every time. So that's really personalized medicine. And in order to do that, we have to develop new technologies like this that allow us to separate the good cells from the bad cells. In order to do that, it's not just cool experiments, it's also a lot of data analytics and a lot of modeling. So you can have really strong correlations between the attributes of the cells that you can measure, the attributes of the patient, and then the clinical outcome that you expect, so that every cell that you put into the patient is the right cell for that indication. So that's what we aspire to, and that's what we're continuing to work towards. So I'll just close by saying, well, what's next then at MIT and for this kind of research and this kind of approach throughout the world? So what's next for us is working closely with our partners in Singapore through a new team that was just launched earlier this year. It's not quite a year old. And that's SMART, which is the Singapore MIT Alliance for Research and Technology, a long-standing program and collaboration between the government of Singapore's National Research Foundation and MIT faculty and researchers and students. This team that I co-lead is called SMART Camp. So we're all campers. And the CAMP acronym stands for Critical Analytics for Manufacturing Personalized Medicine. So it's very focused on the manufacturing and the production of medicines that could work if you knew how to manufacture them so that you have the right medicine for each patient on time and at an affordable cost. So it requires a big leap from growing cells in a petri dish, which was appropriate for basic lab study, to getting cells to actually survive in really complex cell therapy target environments, like a brain. Another way that we're taking this out is not just to focus on the research technologies, but also on the education of the new kind of workforce that's going to be needed for that approach. And so there we have worked with Manufacturing USA. This is a national initiative to develop workforce training, as well as collaboration with companies who want to adopt these new technologies and need to de-risk them in order to bring them as part of the FDA-approved product lines, for example. So the last bit I'll show you here, just a little bit of teaser and eye candy for where this is going. I've mentioned the brain a few times. The brain is your squishiest organ, right? It's very compliant tissue, it's very complex, and most people study the neurons. And when you think about the neurons are long fiber-like cells. They play a very important role in conducting signal from point A to point B. But what we wanted to understand, and here this is two women in my lab who've worked very hard on this for a number of years and now bringing it towards a commercialization path, is what if I could make an artificial neuron? It doesn't do everything a nerve cell does, but the axon, this long part that I'm showing you here, what if I could get the geometry and the mechanics of that right so that I could watch this really key process that is a major part of demyelinating or unremyelinating diseases like multiple sclerosis or Parkinson's. And what if I could actually do that by 3D printing a bunch of these artificial axons so efficiently that I could use this approach for drug screening or cell therapy? So here you see our setup developed in collaboration with Nick Fang in mechanical engineering at MIT, as well as many others over the years. And the outcome that we've achieved here. So this last movie that I'll show you, what you're looking at is these 3D printed artificial axons. They're synthetic polymers. There's nothing biological about them. But then these blue and green blobs, the blue is the nucleus and the green is the cell. And that cell type, its whole job is to wrap around these artificial axons and insulate them so that they're able to actually conduct the electrical activity from A to B, so your nerves operate. When your cells don't do that, that's a characteristic of diseases like MS. If we can watch this 3D process happening, if we can watch the cells wrap around these artificial axons, then we can start to screen that. And here what you're seeing is a little hand of that cell called an oligodendrocyte wrap around that artificial axon. So now we're able to watch this process in the lab and see what conditions could enhance or retard that so that we can be part of the solution as we focus on these diseases that currently have no treatment. So that's what is ahead for us, and here interactions with the Deshpande Center at MIT, as well as several pharma companies have been very instrumental in advancing our thinking on that. Okay, so with that, I will close, and I'm happy to take any and all questions.
Wonderful. Thank you so much, Professor Van Vliet. That was just really fascinating. We do have a bunch of questions. I'm going to take the moderator prerogative and ask a couple before we get to those. I've been spending a lot of time covering the pandemic, so I'm going to ask you a couple of pandemic-related questions. And I'm just really interested with the way that you work and think, and so, multidisciplinary. I would just love to hear your thoughts in the sort of material engineering, biomedical engineering sphere of what, as you're looking at this pandemic, what do you see that could be improved? So I'm thinking, lots of people are trying to come up with different designs for ventilators and for masks and for, killing the virus. There's a lot of effort being spent on the kind of cell level analysis that you're doing in the spheres that you're talking about, but they're looking at, how the virus enters the cells and replicates and so forth. So I'm just curious what you've been thinking as you've sort of been watching this pandemic, that if you had, kind of unlimited time and the ability to sort of tackle one problem or two problems, how would you do that? And what would you tackle?
That's a very good question. So what I am tackling now is being part of a very dedicated team of people figuring out how to help MIT ramp up its research again so we can have more people contributing to those ideas and solutions. How we were in real time developing manufacturing solutions, including face shields that were designed and manufactured at MIT by MIT, new approaches to shared design of ventilators so everything is open source. So I've been able to assist in aspects of that through my administrative role. But I think what I've come to learn, we're all learning about this particular pandemic. I'm fortunate to have worked in Southeast Asia where this concept is not new. And so the idea that you would have to adapt your human behavior and your human work practices and have layers of risk mitigation we're comfortable with that. It's something we've been through before, but not on the MIT campus, not in the US, not in this way. So I think the two things that I think the most about right now is how do we all get comfortable with a long phase of adaptive recovery as a society, that we'll do things differently for a long time, but we'll be better prepared next time. And know that it's not just one thing. It's not just testing. It's not just contact tracing. It's not just face coverings. It's and. And an analogy that's been shared with me from one of my physics colleagues at MIT is when you think about radiation exposure, you always mitigate your risk. More protection is always better because the risks of exposure are known to be bad. So why not do more if you can? The second thing that I spend a lot of my free time thinking about is the challenges in having much faster testing and the technologies that are needed to enable much faster testing. Now, there's limitations to all the testing we're doing right now. The PCR-based testing is just telling you, am I infectious right now? Or 3 days ago when I got tested, right? So there's a speed problem there. And the idea of antibody testing has a high false negative, right? So, and it's also not as fast, but more importantly, not as accurate as we want it to be. So I think that as we develop new technologies that improve on the speed of that, we will get more comfortable with this being a potential illness that affects some of us without taking down all of society or all of our family or all of our region. And that'll give us more, operational comfort. So that's what I'm thinking about these days. And then I have to say the third issue, which is quite nagging, is this is going to be a new threat in the biopharma manufacturing work stream, right? We knew about testing for HIV/AIDS in our blood products. Now, as we start to manufacture more and more pharma products, we're going to be thinking about testing accurately and precisely for this virus or others like it. And so that will take a focus on rapid analysis, and MIT is strong in that. We were working on that before the coronavirus, this particular coronavirus emerged, but we now have to adapt and get ready for tackling this challenge.
Oh, great. Really interesting. Well, there have been a couple of questions related to how the pandemic is affecting work in Singapore, and also, whether, given what you were saying about their previous experience with viruses like this, with, epidemics like this, are they better positioned, for example, to jump into clinical trials, to be involved in pandemic-related research right now? So those are sort of two different questions, but there are a couple of people interested in the Singapore side of things.
Sure. So I think that's a great question, and it's roughly between 3 and 4 a.m. over there. So probably these questions are coming from this side of the world, unless they're really dedicated listeners. So what's going on in Singapore right now is the initial response and the initial containment of the pandemic was a strong one. I actually left Singapore. I was there for MIT in January, and I left just as the cases were getting, the first 3 cases were being identified in the country. So they had a very fast response because they've seen this movie before, and it was effective for a time. And our labs operated, but we went to a shift mode where we had people coming in for certain shifts of the day so that we didn't mix the shifts, and then that reduced the risk of transmission. If someone in that shift was sick, then that whole shift would not come to work. So we adopted that preemptively, and then Singapore's situation got worse, and they went into what they're currently in now is a circuit breaker mode. That's what the local terminology there for all of the labs in my team are shut down, with the exception of one project that's COVID-19 related. So otherwise, there's no lab work going on, just like my lab at MIT has no active lab work going on, all just data analysis and remote work. And they're positioned to start back up on June 2nd. So that'll be the end of the circuit breaker period for research activity like ours. But we had to have very approved plans, and we were ready. We're so practiced at planning for needing to ramp down and ramp up quickly that by this morning we had finalized our plans for June 2nd. So we're hopeful that we learn from that, and we're trying to take those learnings that MIT has learned in Singapore and also adapt them at MIT as appropriate, but each culture is of course different.
Okay, thank you. All right, we have a question from Frank. Some of you have used your name, so I'll use them when I see them. This is not COVID related. Do you foresee any problems in personalized medicine in the areas of privacy or in the ability for individuals to pay for personalized service? Very good question.
That is a very good question. So in terms of privacy, it's certainly a key ethical concern, and it's not addressed just by saying, well, there's HIPAA regulations in the US, right? So in order to make these correlations, data need to be shared. The question is, can you de-anonymize, de-identify or anonymize the data such that I can connect the dots for you, the best approach for you without giving away too much about who you are. So there is some really good thinking about that, not just for cell therapy development. Most of it rests on the idea of leaving the data in a repository and just querying what you need. So you don't need to take all the data in order to operate on the part of the data that you need to help guide the personalized care of the patient. So I think that we'll continue to make progress on that, but it'll be a persistent challenge, a persistent hacking opportunity, right? To say there are data about individuals, and that's inherently valuable data. In terms of the affordability, that varies obviously from country to country and system to system. It is quite challenging in the US because of the reimbursement system we have for medical care. And it's not tied only to the cost of goods, by which I mean I could make manufacturing of a cell therapy much cheaper than it is now by improving on the process, by perhaps automating aspects of it so that it could be an order of magnitude less expensive to produce that bucket of medicine. But that doesn't necessarily mean in the US it's going to get an order of magnitude cheaper. There's a decoupling between the cost of goods to make a medicine and what we charge for that medicine. So some of that is a government role. Some of that is in some countries a patient advocacy role, and it's complex. I think that what we can do as engineers is make the process as efficient as possible, because if the cost of goods are lower, then the price can be lower, and then share the ability to make these medicines worldwide. So the kinds of medicines I work on are generally going to be made locally where the patient is. The bench to bedside transfer is going to be pretty close because of the nature of the medicine. So we can't hoard these advances. We have to share these advances in a responsible way.
Very interesting. All right, we have a couple of cell biology related questions. First one from Bob. Can you describe a little more about how you guide a cell to develop in a specific desirable way? Is it chemical, tactile, pH?
So that's a great question. It can be all of the above. What we try and do is make sure we have a very defined question. So maybe I'll give you a defined question and an example. I showed you those mesenchymal stromal cells that come from bone marrow. And one of the things that we found out about those cells is that by sorting them using that microfluidic sorter that I showed you, the cells that were both larger in the population, so they had a bigger diameter, and stiffer, and their nucleus didn't move around as much because the DNA inside had stopped moving around as much. Those cells were advantageous for helping people who had been exposed to radiation and their bone marrow was destroyed. These cells, when supplied back to them, helped their bone marrow regenerate and helped their blood supply regenerate. And we had very impressive results in animals, in mice, that you could use this subset of these mesenchymal stromal cells to get 80% survival versus state-of-the-art 20% survival. So that's a big improvement, at least in a mouse, right? But it's inefficient because only 20% of the cells that I grew in that environment were the ones that I wanted. So I had a really fantastic graduate student who said, let me figure out what the cells are secreting And then let me see if I can develop a material that will make all the cells secrete more of those key factors, those key molecules. So we called that mechanopriming. She found that by making a polymer of just the right stiffness, sort of a Goldilocks approach, right? And that stiffness wasn't exactly the stiffness of bone marrow, but it was in that range, that she could make all of the cells secrete the molecules of interest. They weren't differentiating, they weren't becoming bone cells or blood cells or anything like that. And the reason why is that they were engaging that material, pulling on it. It affected how they actually transmitted information to their nucleus that affected their protein expression. Right? So we could change the mechanical environment, the cells pull on that mechanical environment, and we can iteratively figure out how to actually control their protein production through that mechanical environment. That took years, so that's not a fast approach, but we learned from that how to get faster and faster in that approach, which is called mechanopriming. It's much more common to change the chemical environment, put different vitamins into the solution, which is called media. You can do that, but it tends to be very, very expensive because of each of those molecules, especially when you want to get into an FDA regulated product, each of those molecules has to be specially manufactured. So we try these approaches that we think have a path toward higher affordability because it'll impact more patients.
Very interesting. Okay, another cell biology question from Claude. Is separating the cells by size as accurate as separating by RNA expression, as accurate regarding cell function?
So that's a great question. I would say not always. In that example that I gave you, we could say that if the cells were within a particular range of 5 micrometers in diameter, we could tell you what the RNA expression, we could tell you what the protein expression was going to be. That once they had committed to that phenotype, it wasn't because the size was special. It was just that the size was a correlate with their phenotype, with their function, right? It could have been that they were blue and their protein expression was what we needed it to be. So we measured in that study about 20 different attributes of the cell, the size, the shape, the deformability, everything we could measure. And this was the strongest correlation we found. But it doesn't mean that size controls protein expression. It just means it's a correlate with protein expression.
Okay. We have a lot of questions. I think I'll be able to get to maybe 2 more. Okay, one is, your work is so omnidisciplinary. How much hope do you have that machine learning will change this work in the next decade? I'm guessing you're probably already using machine learning, is that right? Or
we are, although we're not expert in it. So we use aspects of data reduction, so approaches that are simpler than machine learning because machine learning requires a very, very, very large data set to be effective in the kinds of things that we do. So we're still gathering data to have enough for machine learning to be useful. And that's a big focus of what we're doing in Singapore at MIT right now. So we use approaches like principal component analysis. Can we figure out what are the approaches that show the maximum variance or variation in our data and learn about those extremes? But I do have high hopes that machine learning or other approaches to predictive data analytics will be a big part of turning cell therapy into predictable, useful medicine. Absolutely.
All right, and one final question, but we will be keeping this Q&A box open for about 15 minutes after the session ends, and those questions will be forwarded to Professor Van Vliet, so don't worry if we haven't gotten to them. But I love this question as one to end on because I think anybody listening to you and reading your biography would have a similar question. I bet you have advice on time management with all your duties, so please leave us with a few pearls.
Yeah, I'm not sure I'm the best person for advice on time management. I do manage my time very carefully because I'm really, as you can probably tell, very excited about each of the things that I do. I really enjoy them. So I have to make sure that I fit in time for each of those each day. So I tend to be a very scheduled person. I know the whole week before it happens in terms of the hours I'm going to spend at work and in terms of the hours I'm not going to spend at work, right? They have to be scheduled too for me so that I go for a run or I make a nice dinner with my husband. So I tend to be quite scheduled. That makes me feel comfortable. But I have big chunks of my day, believe it or not, that are called no meetings, because one of the professors at MIT when I was a graduate student years ago said, you have to leave time every day to just be by yourself and think. And I have always taken that advice, and I think that's part of why I'm able to connect the dots, is I do take some time when I don't have meetings each day, and that really helps me rejuvenate and try and connect the dots.
Well, that's a great note to leave us on, very inspiring. On behalf of the Alumni Association, thank you so much for tuning into this faculty forum online. And big thanks to Professor Krystyn Van Vliet for joining us today, spending your valuable time with us. It was just so informative. Alumni, people listening, we are going to be sure to forward all the questions, as I mentioned, that are still in the Q&A to our speaker. And you have another 10-15 minutes to pipe in some more questions. A reminder that this broadcast will be available on the MIT Alumni Association YouTube channel within a week of today. If you're so inclined, you can tweet about today's chat using the hashtag MIT alum. You can send follow-up questions or feedback to alumnilearn@mit.edu. Thank you so much again for watching, and have a wonderful day.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.